our motto is a dialogue with the living, and we present a new approach to the harmony of life. What we call the living, is it made of molecules only, as biochemists seems to believe it? For them, we seem to be just a bunch of molecules without anything else. But these molecules, how do they assemble to form all components of living beings? And these components, how do they harmonize themselves with their environment? And the atom, what is it really, the famous atom? Max Planck, who I will comment on his work later on, said toward the end of his career in a conference in Florence in 1944, having devoted my life to the most rational science, matter study, I can tell you at least that one thing from my researches on the atom: matter as such does not exist. And what if all scales in the living were correlated? Communication by scaling waves between species with their genetic patrimony, ground, terroir, environment, farmer, his implication on his know-how. What if all that was communicating? Is there a hidden harmony, or hidden, not even, we can see it if we just look at it in nature. Albert Einstein, I will comment later on his contribution to that, also said, humans, vegetables, cosmic dust, we all dance on a mysterious melody played at a distance by an invisible piper. So, what is our mission at Genodics? It is, as Pedro mentioned it in presenting himself, to develop and market the applications of genodics, science of waves associated with the expression of the genome. I will obviously detail that later on. Our proposal as Genodics is a new approach of the living, respecting the subject dimensions of all its components. This is resulting from the work of Joel Sternheimer in theoretical physics and its applications in biology. So, what we see now, the origins and principles of these scaling waves and genodic science, the principles of that science, general applications of that genetic science, and more specifically, applications in agriculture. So let's go from the origins. How did research evolve? First, from chemistry to quantum physics. Matter appears to be made up of atoms and molecules at our scale. Chemistry explains their interactions by the exchange of electrons. We presently know 118 various atoms, which are classified in the famous Mendeleev table, also named the periodic table of the elements. I suppose you all know that. Atomic physics has taught us later that atoms are themselves made up of elementary particles. In addition to the electron, we find the neutron and the proton, the photon, but also fermions, leptons, bosons, quarks, muons, gluons, tachyons, leptons, hadrons. In all, more than 100 elementary particles, more or less stable, have been discovered. Quantum physics explores the foundations of these elementary particles and their interactions with vacuum energy. What we think as the vacuum is in fact full of energy. It is occupied by many different kinds of waves, which are forms of energy. In 1900, Max Planck a German theoretical physicist, explained that that different atoms and molecules can only emit or absorb energy in discrete quantities. So the energy is not a continuous quantity. He explained that the smallest amount of energy that can be emitted or absorbed in the form of electromagnetic radiation is known as quantum. This work laid the foundation for quantum theory, and that won him the Nobel Prize for Physics in 1918. This was the origin of all what followed in quantum physics. Planck called the packets of energy quanta, and he was able to determine that the energy of each electromagnetic wave is equal to, given by the equation, E, the energy, equal hv, which actually physicists say h bar, v, where v is the frequency of the wave, and this famous h bar, the Planck constant. That Planck's constant is the elementary quantum of action, and his value is known in meter kilogram second, with the number that you can read there. That's a very, very, very tiny quantity of energy, but it is essential to understand that from the present knowledge, energy is not continuous. It can only be emitted by multiples of that very tiny quantity.
That is the discrete idea.
Yes, yes, it is the discrete concept of the energy. And what we see as matter is nothing more than an assembly of wave vortices. If we could see it at the elementary quantum level. So let's detail this concept of wave-matter duality. Each elementary particle can be associated with a wave. This has been known since the beginning of the 20th century for the photon, which appears either as a particle of light or as a light wave, depending to how you look at it and the kind of experience you are doing. Albert Einstein was the first in 1905 to propose an explanation of the photoelectric effect, which was already known from the end of the 19th century, using the concept of the light particle, the photon, and that of the energy quantum initially introduced by Max Planck. And obviously, he is best known for his theory of special relativity in 1905 and his theory of gravitation known as general relativity in 1915. But this is not these theories which allowed them to get that Nobel Prize. This was his work in electricity and quantum physics. So about Einstein, one last point, that everyone is familiar with the famous equation E equal mc square, which established the equivalence between the mass and energy of a system. This will be also useful to us in the following discussion. So To continue with this wave-matter duality, Einstein has proposed that the duality for the photon. And in 1923, Louis de Broglie, a French theoretical physicist, proposed to generalize this wave-matter duality, telling that a wave can be associated with each elementary particle, but also with each atom and each molecule. And he showed how to calculate the frequency of the wave associated with each quantity of matter based on its mass. For this, he was awarded the Nobel Prize in Physics in 1929. And this is the famous equation given by Louis de Broglie. So considering that every particle can be explained as a wave, how to calculate the frequency of that wave, it is just with that formula which combines the works of Einstein, E equal mc squared, and Planck, E equal h nu. And the energy of a particle and the associated wave being the same, de Broglie just, I simplify, but it just considered that mc2 equal h nu, and the frequency being the inverse of the wavelength. He was able to write the equation here, lambda, the length of the wave, equal h on p, which means if you detail with the speed factors, the speed of the particle v and c, the speed of light, the formula for the is for lambda h on mv and root 1 minus v square on c square.
May I ask something, Michel?
Yes.
The p, the quantity of the movement, what do you mean?
Yes, p is the movement quantity, which is detailed after that with mv and.
But what is that? I mean, quantity of movement. I would think that is
the quantity of movement of the particle. This is the best expression of the energy of the particle. That quantity of movement. This is a basic concept of physics.
In Nederlands noemen we dat momentum.
Yes,
okay.
it is.
Linked
with the momentum of the particle, yes.
Yeah, okay.
Yeah. And this equation was the basis of the work of, for one of the basis of the work of Sternheimer that we will see now. So, this is Joel Sternheimer on the photograph when he was a student at Princeton in United States in 1917. But before to go to be sent to Princeton by Louis de Broglie, in 1966, in his thesis for a doctorate in theoretical physics, at the age of only 23, he demonstrated that the waves associated with the most stable elementary particles, and there were about 80 at that time, have frequencies whose ratios are musical. What does that mean, musical ratios? This means that they are simple fractions, like in all musical scales. You know the octave, which is the progress of 2. You know the most harmonics ratio, which are the tierce, quarts, and quint, which are the ratios between the first numbers, 2, 3, 4, 5. The ratio between these numbers are the basis of harmony in music, and we work with even without to know music. We all feel that it sounds nice when we hear these waves with these ratios. And so he calculated the masses of the basic elementary particles were known, and he calculated the frequencies of all these 80 particles by applying the equation of the relation given by Louis de Broglie. And this is the figures that he got, showing that these particles, the waves associated, are have a regular distribution, and they group by packets, you see, and the packets have harmonic relations. If you calculate the mean frequency of these packets, you will find musical harmony. And this convinced him that there is a hidden music in the in nature at the most basic element, which is these elementary particles which interact with the energy of the void. I mean, so. And in addition to that, in order to characterize relationships linking elementary particles together, he predicted by calculating the existence of scaling waves, linking all organizational levels in matter. He guessed that these particles having their own frequencies in harmony with others, when they interact, they will also interact with all above levels, which also have their own personality. So all these mathematical beings have properties which do not rely on their own expression only, but also on those of other beings of the same time. This is what he wrote in 1983. So this was the theoretical basis of the first discoveries that he made during his doctorate thesis, and after that working at Princeton. But after that, he came back.
Michel, yes. Can I ask a question? There is a question from Annette. What do you mean with scaling?
Yes, scale. Well, it may be translated either by scale waves or scaling waves. Well, I prefer the translation scaling waves, which actually in French, Sternheimers told onde d'échelle, which is scale waves. This means that a scale is, you know, as in music, the music scale, but it is the multiples which link the frequencies of these tiny elementary particles and all atoms, molecules, and all the creation above. And when we translate it by scaling waves, this means a more dynamic process in which these elementary particles waves support and impulse the assembly of all above levels, you know, and the above levels are not anywhere. There are also levels, specific levels where will appear ranges of frequencies when you will find atoms, other ranges where you will find molecules, other ranges where you will find components of the living or of the matter aggregate, you know, and so on and so on. This is a construction beginning with these elementary particles, and by successive associations of these elements, you can go from the elementary particles up to the a group of galaxies, but all that is not has also a discrete structure. It is not continuous. You have specific levels for each combination of the underneath elements, you know.
So like the discreteness of energy quanta,
yes.
You also have a kind of discrete system of waves that
yes.
cannot take every wavelength, but is also discrete, and they are kind of standing waves, but they multiply at every level.
Exactly. They are standing waves which condition the emergence of all the above levels, you know.
Yeah.
Like the scales on a piano.
Scales of what?
On a piano or whatever instrument.
Yes, like scales on a piano, obviously, yes. And harmonics. The key word is harmonics. And actually, still nowadays, the theoretical physics does not consider harmonics and the existence of harmonics and their importance in the structuration of matter. And Sternheimer showed and insists on the importance of these harmonics in the comprehension of, in the understanding of all the levels of matter and all interactions.
The melody of the universe. It's called by one of the people who inspired us. He always talked about the melody of the universe.
Yes, this is, that's it. There is a hidden melody, or so many melodies actually, and infinite number of melodies, each level having its own melodies.
Yes.
And then,
Annette, is
how
that okay?
did...
No, yeah. Please continue.
Yeah, it's okay.
Yeah. And then how did Sternheimer went from theoretical physics to biology? Actually, in the early 80s, friends of him, which were biologists, encouraged him to see whether such harmony could be found also in living organisms. He had shown that the ordinary matter, say, has this basic harmony in particles, atoms, and molecules, but what about the living? And he began to look at books of biology, and this led him to look at a very fundamental process in the functioning of the cells of all living organisms, protein biosynthesis.
Yeah.
As you probably know, proteins are molecules that control all the processes in living organisms. All the cells produce them according to their needs. They are all made of a chain of the same 22 molecules called amino acids. There are billions or milliards of proteins in the living world, but they are all made on the same tiny bricks named amino acids. What are the roles of these proteins? There are many. You can find protein with specific names, you know, for catalyzing enzymes and biological catalysts, for transporting elements, hemoglobin, which takes along the oxygen of the lungs toward the organs. Communication, for example, hormones like insulin or flowering. Indication, information, chemotactism. Recognition, like the immunoglobulins. You have proteins of structure, like the extensin, collagen, or the proteins of the cytoskeleton, muscles of the skin, of bacterial whip. So, I mean, you see that whole world proteins, and they are the elements which will drive all these processes. Let's have a look on the protein biosynthesis. So you have a, let's consider just a plant. It will be the same for an animal. You have the whole plant, and if you look at with a microscope, you will see cells, and in the cell you have the noyau. I'm thinking well, the cytoplasm, and well, we see it again. The central part where you have the
how do you call it in English?
Nucleus.
We call it the cell kern.
It's nucleus.
Thank you.
We'll see that. I will have
nucleus.
a small video in English explaining that.
Yeah, it's
the nucleus. Voilà, that's the nucleus. At the center of the cell, you have the nucleus, and which contains the famous DNA. And that DNA has all the information for building the whole organism. We will see how that works in that video coming. Where is that video? Yeah, okay, here. I have to click. I have to click here, and the video should start.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
That's it. So this video is quite interesting. How can I get out of it? Well,
you click on the little cross at the right. Yeah.
Right, okay. So as we saw in the video, the first step for preparing a protein is the transcription done in the nucleus and producing that famous messenger RNA. And after that, when the messenger RNA is taken by the ribosome, the translation is done, associating one by one amino acids, which are brought to the ribosome by transfer RNAs. We will, this is what biologists know, but Steinheimer, looking at that, had a complementary view that I will detail. He was interested by that, which is known by biologists, but saw it in a slightly different way. And this leads us to the principles of that genetic science in biology. So we consider wave phenomena specific to the synthesis of proteins. Amino acids, basic components of proteins, are molecules which in specific conditions can emit a wave. This is application of the duality, matter-wave duality. And these waves may have a regulation role in biology, complementing well-known biochemical processes of protein synthesis. Let's see how that works. Here you see transfer RNAs, which are in the cell, first hooking amino acids. What I didn't tell, but it is that every cell in every organism has a stock of amino acids. How do they get them? That's by feeding, whatever the type of feeding of the organism. The sap or the blood will bring to the cells the amino acids that they need. And so some amino acids cells are able to do by themselves, but most they have to get from the feeding. And inside the cytoplasm, so you have these available amino acids, and which are taken by transfer RNAs, whose role is to bring them to the ribosome. So this is the ribosome, and when the transfer RNA arrives on the ribosome, actually the amino acid emits a wave impulsion. Let's see how this can happen. So the ribosome, as you see, is a sort of workbench who's specialized in assembling amino acids in the specific order indicated on the messenger RNA, and in order to produce the corresponding protein. And so to assemble these amino acids in the right order, we show in the video that there is a very specific system of codons on the messenger RNA and anticodons at the basis of the transfer RNA, which allows at each step only one amino acid to be accepted by the ribosome, just to have the right order in the amino acids. But analyzing this process, Joel Steinheimer realized that each amino acid carried by transfer RNA, when it lands on the ribosome, undergoes a sharp slowdown. I mean, its speed, he was in a Brownian movement, you know, and his speed is strongly reduced. And the direct consequence of this is an increase in the phase amplitude of the wave associated with it. Just because, you know, the energy of that amino acid is not changed, but if if it slows down, then the amplitude of the wave is increased. The dimension, the phase dimension, actually, in physical terms, is increased. And by calculating that, he showed that at that stage, the dimension of the phase of that wave overpasses the physical dimension of the amino acid, and so the amino acid is, during the very short moment, in a position to emit a wave impulsions, and which frequency, obviously, with the frequency associated with its mass. So when here you have the red amino acid plugged on the ribosome, and his amino acid will link to the previous ones, and the blue one is arriving, and in the stage of emitting the impulsion, wave impulsion corresponding to its mass. So, and after that, you have a green one approaching. So you have a sequence like this. The green one is approaching and emitting his wave, and then a violet one, and a white one, and so on. You see, a green one. So you have the section of these of the wave impulsion which are emitted. So this was the idea of Sternheimer, applying the basic wave matter duality principles and the equation of de Broglie, allowing to calculate the frequency and the dimension of the wave associated to their mass. These melodies associated to the synthesis of a protein are, of course, inaudible. The calculation gives frequencies actually of the order of 10^25 hertz, you see? So far above what we can hear and even reproduce. So to be able to listen to these succession of impulsions, wave impulsions, Joel Sternheimer had the idea of transposing them into an audible scale. I forgot to tell you that he was a musician. Actually, he has a short career as a singer in parallel with his studies under the name Évariste, a glimpse to the famous mathematician. But according to the rules of musical harmony, all you have to do is go down the octaves to approach the audible range. Mathematically, this is not complicated. To reach a range of frequencies between 200 and 800 hertz, which is very audible for everybody, you just have to go down 76 octaves. For a mathematician, this is not complicated.
How many did you say? 76?
76 octaves, yes. To go down from the original frequencies associated to the masses of these amino acids to audible scores, notes, you know.
Yeah.
And due to the fact that these frequencies are in harmony at the original level, they still are in harmony 76 octaves below, because the shape of the signal is preserved. Going down the octaves, you keep the intervals between successive frequencies. They do not change, and so you can recognize the shape of the melody. That exactly like with every song, you know, when you sing something, you don't know the frequencies you are emitting, but other people will recognize the sequence of not the notes. Everybody will recognize the intervals. This is what we feel, recognize, memorize, and can emit at our turn. So this is actually the frequency is not the most essential part. It's necessary to have the melody, but the information is between the notes. This is not the frequencies which are involved in these processes. they are the intervals between the frequencies. So this is much more subtle when you understand that than pure interactions with frequencies. So everybody is speaking about, yes, frequencies, resonance, and so on. Okay, that exists. But there is also another phenomena associated not to the frequencies and direct resonance, but to the intervals, to the shape of that very specific signal. And Joel Stelheimer gave to these particular melodies, protein melodies, the name proteodes. So, what about, let's comment about these very specific amino acids frequencies, which are the base for these proteodes. These amino acids, actually there are 20, which are the most frequent. There are 2 additional, which are very rare. They are synchronized on 10 frequencies, and this is the genetic code. That's given by the calculation. These 10 frequencies broadcasted at cellular level are musically transposed in the audible range, correspond to 10 notes of the 12-tone equal temperament. 10 frequencies for 20 amino acids. Then some amino acids have the same frequency in this genetic code. During the synthesis, homologous sequences of notes will resound together and foster the pairing of codons, anticodons. Actually, Stelheimer supposed that that melody is not for nothing. It has a role in facilitating the building, the assembly of these amino acids to build the protein. And this was just an idea, but which was verified later on when we were able to experiment and to measure the impact of such melodies. This is what I will comment later on also. So a proteode is a suite of audible frequencies calculated with the genetic code and applied to the structure of a protein, the chain of amino acids composing that protein. Via these scaling waves, the melodic message carried by the proteode may resound with the melody which accompany the process of synthesis of the corresponding protein at cellular level, thus generating an effect of stimulation or inhibition, which means that the proteode corresponding to one protein will stimulate the production of that protein. And we will verify this later on. What we demonstrate is that the stimulation type resonance of that proteode produces an increase of the synthesis of the corresponding protein. So, in the process, amino acids may have a wave behavior. We saw that. So you have here a ribosome going on a messenger RNA and a succession of impulsions of waves emitted during the process. So what is that genetic code? I told you that the calculation gives 10 nodes, and you have them here on that table. So glycine is low A, alanine C, serine E, and so on. You have all the amino acids here with the 10 nodes of that genetic code. And then the basic work to compose a proteode, the first step, I would say, that is, you won't get the fully fledged proteode like this, but the first step is to convert the sequence of amino acids for each protein in a sequence of notes. You can see an example here on beta-tubulin. You have the sequence of letters corresponding to the amino acids. There is an international code allocating one letter to each amino acid. That's quite easy. You have 26 letters, so the 22 are enough to characterize the 22 amino acids. So this was codified. So you can find in the databases the sequence of amino acids for billions of proteins nowadays, and this knowledge is increasing day after day. And there is an international agreement making free this knowledge for all humanity. So laboratories are implementing these databases with their discoveries day after day, and all that is available for everybody. So when you want to know the structure, the composition of protein, you just go to these databases. There are 3 in the world, and get it. So that's very easy. And I told you that these frequencies are in harmony, and here is the scale. I just forgot to tell you something, because here you can see 2 notes for each amino acid. It is that I told you about the direct melody associated to a protein, to each of its amino acids, but Sternheimer, having felt and analyzed, found effects of these proteodes, I will comment on that later on, found that it would be useful also to do the opposite. If you consider that listening to a proteode may have an effect on your own biology, sometimes you might need just the opposite, if not to stimulate a synthesis, which may then be in excess, but also to reduce the synthesis. And actually, he had the problem when he first listened to the melody associated to I just a protein to relax yourself, to erase pain, you know. And
painkiller.
Pardon?
Painkiller.
Well, not that protein that you produce naturally when you make efforts, and when you have an injury.
Endorphins?
Endorphin, thank you. Endorphin, yes. Okay, and this is produced naturally in organisms, and he had this is quite nice to listen. It's like a baby song, you know, to relax, and he had listened it a lot, and then found himself in a kind of apathics, you know, too much relaxed. And then he thought, well, is there a manner to avoid that, or to reverse the process? And he discussed with his neighbor, who happened to be a biologist, Vincent Bargouin, and who just told him, but if you know to do something in one way, you can produce a melody. Can't you reverse it to have something in phase opposition? And this gave him the idea that he might just return the melody, to return the intervals between the notes. If the direct melody is la la la, you just do la la la. You just take the symmetric melody, you know. And doing that with that endorphin, he realized that he felt that this was liberating him of that too strong effect of the direct listening of that proteode of the endorphin. this is how he constructed the opposite code. And on that scale, you can see the two codes, the direct one and the opposite one for all 10 groups of amino acids. So with that knowledge, we are able to produce for each protein 2 proteodes, the one in stimulation and the one in inhibition that we can use. You can reinforce a process or reduce another one, but you can't really you cannot really do all that you would dream of. You have to be in accordance with the real needs of the organism, which with the scaling waves has also a word to say in what you do, you know. But this is quite useful, and this is what we apply, and we'll see in the following pages.
But in the may I still ask a question about the previous graph, because these pink amino acids in the center, they have only one tone, so you have that's no inhibition or stimulating.
Yes. This one is just in the middle, just in the middle, and so the frequency is the same in the two scales. Yes.
Okay. I see.
But Marijke, because of the intervals are important.
Yes.
It's the interval between the yellow and the pink and the brown. That
exactly.
Is not the position of the node itself.
Yes, not the position of the node is important. It is exactly the intervals which are important.
And then a question, Michel. There are 4 pink and 4 yellow and 4 brown.
Yes.
And how do this 4 yellow, for instance, know which of the 4 will attach into the ribosome?
They don't know. This is just the effect is not. This is done by the codon-anticodon structure, you know, which will make the final selection. The melody will only condition the approach of these transfer RNAs, but the impairment is done by the associating codons with anticodons.
Yeah, got it. Thank you.
Yes. An interesting consideration is about the repartition of the pitches between frequencies. They follow a musical consonance law, you know. If you consider the spreading, all the potential intervals between the 22 amino acids, this is an average on 2,500 human proteins. And these pitches, these intervals, are grouped exactly in the same way than music, human music. You see, you have at the center the unison, which is the most used pitch, and you have the very low the tritone, which is not harmonic, and so on. You see, you have all these potential pitches between all these notes, and the spreading for an average of 2,500 proteins is the same than the spreading in human music. That's an interesting consideration. Let's have a look now on because here you can see the scores for protein musics. If you can read music, you see the transposition between the series of letters corresponding to various amino acids and their notes. Actually, these proteodes are real musics that you can write on a score. And these suits of broadcasted frequencies participate in the regulation of the synthesis of proteins, and which is surprising, but reproduced into the audible range, they have physiological effects. As I said before, this is just because the shape of the message is conserved. And what you feel at our level will interact through the scaling wave with the micro level of the cell and the process of assembling amino acids to create the protein. That's a discovery. I mean, it was not obvious. Steinheimer didn't know that. He found it by chance, you know. His idea was to listen to this series of frequencies, and then he felt something at listening to that endorphin, and to others also. And after that, he realized that there was also biological effects which were able to be measured. The first discover of that time was the wife of one of his friends. This was in November 1985, and she had a deficit in hemoglobin, and he gave her to listen the melody of the hemoglobin, which he had done, and she reacted strongly, telling that, wow, that's fantastic music. I feel good. Can I listen it again? And she listened it 3 times and said that, oh, I feel much better listening to that. And she had blood tests every week to see how his disease was evolving. And this was a Thursday, and the following Monday, she had recovered the normal hemoglobin level, you know. So she called Steinheimer to tell him that, and he was surprised, and this was the beginning of an interest to look more deeply to that and to try to understand what was going on, you know. This was the first measured effect of proteolysis, which gave paved the road to the further trials and discoveries. So the fact that you have only 10 notes for 22 amino acids will provide you with other homologies. Biologists are looking at homologies between sequences of amino acids, but considering the melodies associated to these proteolysis, you will find homologies of frequencies, of rhythmic structures, and which will indicate potential cooperation between proteins, but thanks to the interactions of their melodies. This explained that when a protein is synthesizing and emitting its melody, it will also inform other proteins that it's doing that, and stimulate proteins which have some common elements, some homologies with it, but also inhibit proteins which are in phase opposition. So this is a fundamental regulation happening in all organisms, just by the fact that when a cell somewhere is synthesizing a protein because it needs it, it will also inform other cells around that it's doing that, and allowing synthesis of complementary proteins or inhibiting the synthesis of others, which are detrimental. So let's see a musical example. Here you have the actin proteody. And I will just quit that presentation for a short being because I was not able to link that to the melody, but I will give you to listen the proteody of the actin, and you might feel that there is something reminding the primavera. Let me escape to that. I will close that window.
Yeah, the Vivaldi.
Yeah, I don't need that. And here is the actin. Okay. Did you felt in the very beginning? La, 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 la. Did it remind you the famous Primavera from Vivaldi? There is a direct correspondence. Without to know biology, by listening that proteody, you may feel that, oh, that I know that melody. And Vivaldi felt it when he composed his Primavera spring melody, and that's it. We have many examples of human musical compositions which integrate parts of proteodes, and this is not only chance. This is the fact that the composer feels the nature around and translate that in sequences of notes, So that's just interesting to see and check. So let's go down. Let's go back to the diaporama. Okay. And just to summarize, of proteodes by cells producing proteins will interact with the human environment, with microorganisms, with vegetal and animal environment, and why not with mineral environment? All that dialogues, in a secret way that we can't hear, but which is real. So we the first biological considerations by Steinheimer began in 1982 or 3. The first feelings and measurements were in 85, but after that there was quite a lot of work to be done to understand what was going on, and a number of friends of Steinheimer aggregated with him to try to experiment that. As Pedro said, he met Joel Steinheimer at the beginning of the 90s, yes, and the first steps were done on plants from in 1992. They were made on tomato plants, salads, and actually Pedro made the first experiments on tomatoes in the backyard of her grandmother nearby Paris, and he was well, he was a student in agronomics at that time. This is why he was interested, and he saw that by giving to listen to the tomato plants specific melodies, he was able to have taller plants or more flowers or bigger fruits, just by testing different proteodes. Other experiments were done in Switzerland, Japan, Belgium, and even Senegal. Pedro went, a friend of ours, paid to Pedro the trip to Senegal to experiment the resistance to drought of tomato plants. And this was quite significant results. Also in 1993, Pedro made the tests of bread making in his school, the ENSMIC, École Nationale Supérieure for doing bread. And he presented his results in technical days and published the report in the magazine of that professional sector. Another interesting test was done on luminescent bacteria by a student with his professor in the University of Metz in 2002. And a series of very interesting experiments were done also on the production of interleukin-2 in cell cultures at the School of Chemics in Paris by a specialized biologist, friend of Joël Sternheimer. More recently, we developed a partnership with Hermès Laboratory at the CY University of Cergy-Pontoise. During 10 years, they made a series of blind trials on pea germination, measuring epicotyl size and protein concentration, and this allowed them at the end to publish, to get the first official publication in a peer-reviewed magazine about effects of proteodes. I will comment to that later on, just after. And last year, they began a new series of experiments of proof of concept at the plant cell level. So let's see, have a look on that work, recent works with that university. We'll come back after to the applications on the ground, but that's the...
Michel.
Yes.
I don't know how it's about everybody, but I mean, we are now nearly 1.5 hours on the way. Maybe we want to have a little break.
Oh, if you wish a little break, okay.
You want to continue? I mean...
No, but it's just up to you. I mean, I can continue.
And people who want to continue put up their hands.
Does somebody wish to have a break?
Someone wants to have a break?
I would like to have a short break.
Okay, shall we take 5 minutes break?
Okay. Let's take 5 minutes.
Toilet and so on. Yes.
Okay.
5 minutes.
Keep the computer on, but let's have a short break.
Yeah, it will run, but doesn't matter.
I will have a drink then.
I'll just pause the recording. Yeah.
Okay.
Where do I stop it? Oh, here.
Okay, let's.
There we go.
Okay, let's go. So let's have a look on the work done with that research team at the Cergy-Pontoise University. We realized that when working with them, we made a research on other teams working on plants and sounds, and we found last year that there were already at world level 2,513 publications on that theme with various approaches, showing a growing interest. There were very little at the beginning of the 20th century, but there is a growing interest in that. And the first results, the first publication done by that team, associated with Genodics, obviously, was in the Cell, in the Helion magazine of the Cell press, linked to Elsevier. And the title was The Diffusion of Sound Frequencies Designed to Target Dehydrins, Induce Hydric Stress Tolerance in Pism Sativon Seedlings. What were the results? You have this was done at plant level by cultivating for 8 days pea seedlings under salt stress and subjected to proto-mimetic sound waves. They're written, Olivier Gallet, the director of that team, forced the term of proteomimetism, not to use the proteody term. But the concept is right. It's a kind of proteomimetics. And the frequencies of these sound waves are associated, as you know, now with the amino acid sequence of dehydrin. They use for this test only one proteody associated to dehydrin, a family of proteins implicated in adaptation to water stress in plants. And they use it in the sense of stimulation on batches on one side, or inhibition on another side, with a test without music in the third room. So they worked in 3 rooms. And they work only with a very short melody, the sequence of the first 25 amino acids of that dehydrin. And you have here the associated notes in stimulation and inhibition. This was done in triple blind on 3 different sites. This means that the person installing the proteoses or the sounds, they didn't know even what kind of sound they had, didn't know what they were installing and potential effects. Then another person came after 8 days to collect the seedlings, wash them and measure the length of epicotyls and the weight of the whole produced matter with roots. And these data was registered and analyzed by the third person, which didn't know what had been done, you know. So, it was completely triple blind work. And in short, the average difference between those having received the stimulation and inhibition was about 37% of additional matter for those having received the stimulation. And so this was very, very significant. I mean, as such, it doesn't have direct application. I mean, except that proteody stimulating dehydrin is very common in plants and has a wide range of use to help plants to resist drought, you know, which is a growing problem nowadays. So
Yes.
it's quite interesting series of experiments. And what is also interesting for us is that that team accepted to work with us at shared costs, which means that we didn't pay them for that, and they didn't pay us for our experiments and our, directions, advice, and equipment. Because we were not able to pay for them with our small budget. And they accepted to do that for science, you know, just for the pleasure of experimenting something really new. This is why it took time, because they did it when they had time between the other programs, paid programs, you know. they inserted these experiments, and it took time to settle a very, very symmetric process. And at the beginning, they tested stimulation against nothing, and well, it was not symmetric. So they found that the best way was to have some kind of music on all batches, and the most obvious that you have the best results was to stimulate on one side and inhibit on the other. This is what was done for tens of series, and reach very high level of significance.
Okay.
Okay.
What?
This is what we see here, the results.
Yeah.
And another work started last year in March 2022. And this one is paid. Thanks to that first official publication, that research team was able to get a subsidy from the French National Research Agency to experiment at cell level. And this is a 2 years program which is going on. This allowed them to recruit 2 researchers to work full-time on that, and we also participate in the research and the basic team also. So there are 6 persons working on that for 2 years. For Pedro is half-time on that, and we have another guy, Victor, 4 days a week, and plus the research team of the MS laboratory. So results are expected for next year. Oh no, yes, beginning of next year.
Okay. And what were the results of the P experiment? I didn't get it exactly, but
You get a real difference between the stimulated and inhibited batches of seeds.
so
You have the P wave frequency distribution on the right, which are clearly different for the 2 series. It's a statistical analysis, you know, and also the detailed analysis. Well, they measured 2 things: the weight produced in the various seedlings, and also the concentration of the hydrin to verify that there was not only the global effect, but also the effect on the production of that specific protein. And this is what was measured on the left, comparing control and stimulation. You can get the publication online. It's free. I mean, to read all details.
Because I mean, compared to how much money is put into genetic modification of plants to stand water stress, and what you are doing for free with results, then
yes, but
what a strange world we are living in. Yeah.
We were not able to get any subsidy for many years, and we are very grateful to that team to have accepted to do it for free, because I mean, we demonstrate on the ground, in the field, but it doesn't have the same scientific value, because there are so many parameters, you know. We are always told by specialists that that doesn't prove nothing what you get on the field, because there are so many parameters that you can't affirm that this is only your music which produces that. It may well be something else. So this is why we needed these laboratory experiments, very well done in triple blind, to show, okay, look at that. That all parameters are mastered, and you get those results. So this proves scientifically, I mean, the reality of the phenomenon.
And there are two questions in the chat. One is that if the effect of the GSM radiation has been measured of the telephones. And the other one is if you could show an example of harmonic dissonance inhibition. Or inhibition. Yeah.
Harmony. Example. You want? I mean, if I gives you to listen the inhibition version of a proteode, you won't feel anything else than with the stimulation. It all depends on your need at that precise moment when you listen to it. If you don't have any cell producing that protein, then you will feel no interest in listening to that. And if it contradicts some biological process in your own biology, you will feel it quite irritating, disagreeable to listen. So I mean, the effects are linked to the real need of the organism. In this experiment, these were seedlings of peas under hydric stress, you know, and so they needed that stimulation, and those getting the inhibition were not happy with that, so they grew less. But
well, my question is, Piet, would it apply for some noxious weeds? For instance, if you put let's say the inhibition sequence on noxious weed in a field, that means that you could just let's say prevent the noxious weeds from growing. Just as an example. Is there some research on it, or has it been looked at? I can imagine if you can apply an inhibition sequence on a certain noxious weed, or let's say a combination of noxious weeds, then you can just, let's say, put it in the field, the certain sequence, and then it can prevent those noxious weeds from growing.
Yes. I will show you examples on many diseases, for example, for which we try to inhibit the pathogens. But what I can tell on a global basis after 15 years of experiments with Genodics and others before, is that the effect of inhibition is smaller than the effect of stimulation. Just because, you know, the stimulations accompanies the process. And so if this is in phase with the need of the organism, whatever a plant or animal or human, the organism will take it and reinforce its activity. But if you disturb it, the organism will try will still try to do what he was doing. So it will resist the melody, the proteode, and this is how we interpret the fact that globally the effect is lower. And when we have to deal with diseases, we always search for complementarities with other proteins. When the organism is able to produce molecules to resist the pathogen, we stimulate the production of these molecules in addition to the inhibition of the pathogen, you know. So the development, we will see the applications just after. It's quite rare that we use only one proteode. In these theoretical, laboratory experiments, this was done with only one proteode just to see the effect of one proteode on a specific model. But on the ground, we always use a combination, and that the high expertise of Pedro to find the key proteins and assemble proteodes convenient to each probiotics, you know.
Yeah. Okay, and the other question about the GSM radiation.
What was the question?
The same lady also said, but it's also possible to ruin the crops. but if you have measured any influence of the radiation of telephones,
No. Oh, I'm personally convinced on the bad effects, of that. But first, there are completely different waves of what we use, and it's a different field. We never analyzed and measured that.
Okay. But if you use the wrong frequencies, you might also ruin the crops. That's what Annette also says. Is that true? Do you have experience with that?
If you use the wrong frequency, what do you mean?
The wrong melody.
Wrong. Oh yes, sure. We have cases where if you don't use the right one, if you use in the wrong direction, or even if you use it too much, there is also a notion of quantity of the duration of and volume of the stimulation or inhibition. If you the experiments proved a long time ago that Pedro noticed that already with tomatoes 30 years ago, that if you stimulate too long, you will have necrosis on your tomatoes, or they will begin to crackle, to lose water and so on. So, our expertise is not to use too long. The usual duration of proteolysis diffusions is between 5 and 10 minutes, and not more. And for important problematics, you can do it 2 or 3 times a day, and that's it, no more. But you may have different sequences at different times, targeting different problematics. But altogether, we never go over 10 minutes for each diffusion.
And here
yesterday, Pedro told me an interesting example. He said, if you apply for too long this music, the plants get tired.
Yes.
And the end result is indeed necrosis.
Yes.
I can imagine that, because you add all the time energy to the plant and it has to handle it. So there must be a limit in what they can elaborate to their health.
Yeah.
And I think also he said that if you have many plants have the same proteins, including weeds, as for instance tomatoes or potatoes or whatever. So every plant that receives the waste will have support in growth, also the wheat.
Mm-hmm.
So that's very interesting. But I think the profit is in stimulating the plant that you desire to grow, and then it will also be strong enough to handle the correct competition of the wheat.
Yes.
Is that correct, or do you have specific melodies to affect the weeds so you can suppress them?
We never try to stress plants. We try to help them.
Even not weeds, what we call weeds, because weeds are also actually just natural plants that.
I think it's very important not to go against growth principles, because not only the plants will suffer from it, but also animals, also ourselves. We also have a lot of proteins that are the same that you find in plants.
Yes.
So that's also another reason why you should not broadcast this music for too long, because people may have consequences as well.
It's true, and we always insist when we install applications with farmers that they avoid to be there every time when the broadcast machine will begin. I mean, listening from time to time for a few minutes is not a problem, but that's the addition of the effect day after day, which may be detrimental. And it depends on their own biology, as I said before. And we know that there is a direct correspondence between the feeling you have when you listen and the effect on your own biology. But for that, you need to be conscious, to listen attentively, and to examine your feeling, because an accident between brackets may happen if you listen without being conscious of the potential effect, and listen something which is not in accordance to your need, or maybe be amplifying a disorder that you have, and it's just a question of attention. If you know that you just have to feel, do I like that or not? If you don't feel any irritation, okay, it's okay. You may let it go. It won't have any detrimental effect on you. But if by paying attention to it, you feel that, oh, I don't like that, then you go away or you stop it.
Yeah.
That's it. But who knows that? Very few people at that time, for the time being. So we had clients having problems with their health, and who asked, why do that happen? And we explained them. And after that, they paid attention to, and not to be there when the music was going on.
Yeah.
No, in short, it's only going to be used well in a positive way. You can only use it positively, and not in a destructive way.
Yeah, you from our experience up to now, we never found that you can kill an organism with that. But you may amplify existing disorders,
Yeah.
Good, thank you.
Okay.
Okay, let's continue. On Cell and Sounds, there are much more publications. The same bibliographical research found over 17,000 publications on Cell and Sounds between 1907 and August 2022. So this is more widely explored. So what are the basic findings and principles of genodic science? First, respect for the subject. The subject, I mean the subject of the living organism. To be effective, that method needs to be in agreement with all levels of the organization of the organism. The more echelons are and resonant organisms there are, the greater the range of the scale wave effect. This allows a better expression of the organism potential in harmony with its environment. It's a wave phenomenon and does not use any molecular input, so no material pollution. And it is also a new key to understanding life by these correlations, musical correlations between proteins according to their associated melodies. Just a brief consideration of on the evolution of agriculture. I consider that there are four ages which are cohabitating nowadays. The old age was hand farming with the workforce of men and women, hoeing, planting, weeding, removing pests by hand, cooperating with the force of nature, including agricultural songs. They felt that already. And for the lucky few, animal traction. Then came mechanical farming with steam and then petrol engines. Farmers felt that they had power to dominate nature and developed deep plowing, increased use of tools, cutting out anything that doesn't comply. And after that came chemistry, assembling atoms, and using agrochemical molecules. So they considered, and still nowadays most biologists consider that plants and cells are communicating via molecules, which is true, but this is not the only communication way. I mean, this is an analog to the post office, but we also developed radio telecoms, and the plants invented them before us. The growth was a question of supply of nutrients and diseases. Facing to diseases, you had to eliminate pathogens, and this is still the mainstream politics in agriculture, eliminating all pathogens with all the troubles that it may cause. So Genodics see the things is the fourth type of agriculture, the fourth era for us, assembling sounds, proteodes, so with waves associated with molecular mass and specific melodies of the living world. Plants are emitting and receiving waves. The growth is a question of optimizing metabolism, and illness is a question of dialogues with pathogens, reestablishing a good dialogue, allowing the organism to live with these pathogens. We consider that it's not necessary to eliminate all pathogens, because maybe if you do that, a worse pathogen will come. It's just a question of equilibrium, balance. Reducing, if the pathogen takes too much room, okay, you have to incitate him to reduce, but you don't need to kill him. He may have some positive use also. So it's just a question of balance between the whole organism and each specific pathogen. So it's a different view on agriculture. Obviously, the other approaches may be used in parallel, but we prefer the fourth one. But we understand that there may be needs from time to time to apply also other knowledge.
Michel,
yes.
In the Netherlands, we have this fundamental debate of the future direction of agriculture, and one of the directions is what is called nature-inclusive agriculture. And I think this fourth family, your Genodics family of farming, is very fundamental in supporting this nature-inclusive approach.
Yes, sure. We are in harmony with that also.
Yeah, exactly. Thank you.
So let's see now the general applications of the genetic science on the ground, in the field. There are types of applications. One at the scientific level, predicting protein functions and studying homology between proteodes. This allows to predict regulation possibilities and looking to avoid, to prevent potential side effects. We always do that before to implement some groups series of proteodes on the problematics. We have this first bench research. And after that, we may regulate biological processes while respecting creatures and their environment if we have done the first step before. And this allows to take into account the different levels of living organisms. Our products and know-how, we produce the sound sequences tuned with amino acids composing proteins. We set up applications based on the diffusions of assemblies of these sequences, and we control biological processes by stimulating or inhibiting the synthesis of proteins. These are services, but we also have products which are the boxes used to broadcast these sequences. So we have, for the time being, applications in agriculture, breeding, agriculture and breeding. The others are experiments, but no systematic applications in agroalimentary, environment, energy, pharmacy, health. We dream of that, but we are looking for additional financing to be able to begin with these other sectors. So let's see what's about in agriculture. We have quite a diversity of applications. You have here views of various experiments, and I will comment on all these. What we do is stimulating the growth of plants, limiting and saving in inputs, reducing the use of chemicals, water, energy, helping plants resist to drought, also resist to cold, preventing and curing diseases, and helping to improve qualitatively and quantitatively production. Well, that depends on the type of on the conditions of production. In where our experience showed us that in greenhouses, modern greenhouses where all factors are mastered, proteins won't add much. You will get at the best 5% more, and not always. I mean, they don't need proteins. They have other means and other problems, but when we can really have measurable impact is outside and in real conditions. When plants meet difficulties in their environment, then we can help. If everything is done artificially to master all parameters, proteins won't add anything. So just a rapid view on breeding applications. We worked on increasing in quality and quantity the milk of cows, goats, and sheep. We work on the treatment of diseases, on young porks, post-weaning bacterial blight, oysters, reducing the impact of oyster virus, the herpes virus of oysters on young oysters. We did that in partnership with a laboratory in Caen, in Normandy, with very good results at the laboratory level, but we were not able to find the financing to implement on a broader scale. And at that time, this was 10 years ago now, producers were not interested anymore because they had found how to produce healthy oysters abroad in Ireland and bring them in France, just changing their passport and selling them as French oysters. But the prices had been multiplied by 3 due to that crisis, that virus crisis, and they didn't want the prices to go down with an excess of production. So they were not interested by our technique which might have reduced the mortality. So that's it. We also worked
Michel,
yes.
There was a question about the quality of milk.
Yes, we see that. We see that on the quality of milk with an example in Roquefort. The milk by oats with milk used to produce this famous cheese, Roquefort. You'll see the result. I will comment on that. A practical example. We used Pedro prepared sequences of proteodes lasting 8 minutes, designed to stimulate the production of lactation protein, prolactin, and make milking ewes easier. After installing a programmable mini audio system, this sound sequence was broadcasted before each of the 2 daily milkings. And first, they had a 3-month trial on the first group of 48 ewes, identified with daily monitoring of the quantity of milk collected and its composition with protein rate and butter content. After that, the test was extended to the entire flock. It was a small producer with 250 ewes. This began on 25th April in 2016 with monthly continuing monitoring and weekly monitoring of the usual milk quality factors. And broadcasts were stopped 2 months after, on 15 June 2016, so as not to interfere with the insemination of the ewes in the sheepfold. And these are the results on that stimulating lactation in ewes. You can see the curve in blue was the year before, the evolution of the production the year before. In red, you have the beginning of 2016 without diffusion of proteodes. Then the yellow part, the diffusion of the first group, and after that, the green part on the whole group, and the last brown red curve is when the diffusion were stopped. And then the growth curve became flat. So we see on these results of analysis that the production that year in 2016 began at a lower level than the year before, but then went over during all the period of diffusion of these lactation proteodes, and then the effect stopped when the diffusion stopped. So this was on the quantity. I don't have the result on the quality, but it was measured that they had a net improve in the quantity of proteins in the milk of well, between 15 and 30%, and actually the producer was happy with that, but he didn't want us to communicate on that because he was not doing his cheese by himself. He was selling his milk to the cooperative, doing the cheese, and the cooperative was paying according to the concentration rate of proteins and grease in the milk, you know. And so the higher level he had, the better price he got. And he didn't want the cooperative to know what he was doing, and also the other producers doing the same, because the cooperatives would have changed the price rate, you know. So and after that, they didn't want to continue to pay us for that, and so we stopped. Well, they kept the equipment, and we don't know, but we believe that they continued. But it was in remote parts of the central mountains of France, and it was too difficult for us to go there to control, and so we let them. But this was not enlarged to the whole production sector at that time.
And you put the music in the meadows, the equipment?
No, not in the meadows. We did that just. We didn't try that in the meadows.
In the stable, you put it.
Yes, in the stable only.
Just before milking was applied.
Yes, just before milking. Yes.
Okay. Yeah.
Another interesting experiment was on the prevention of post-weaning bacterial infections for young piglets. Usually, in the days following weaning, piglets are highly susceptible to infectious agents, and particularly the famous Escherichia coli bacteria. And to prevent this bacterial disease and improve the well-being of piglets, pig producers in the Oise region, north of Paris, began applying the Genodics process in April 2010. And here you have the results. It was used to you know, he got batches of 200 pigs every 2 months, and well, you have the numbers of piglets that he had. Here you have the first series was only 112 pigs, the second 196, the third 2035. These piglets were produced by specialists having pigs to do that. And then his work was to feed them and to grow them, you know. Basically, he's a producer of cereals, and he add value to his cereals by feeding piglets, you know, and then sells the mature pigs. And the young piglets, when they arrive, they leave their mother and are transported and arrive in that new place. They are highly stressed and will get all potential disease which are around, you know. So he was used to lose to have death of piglets, and this death was completely suppressed just by giving specific proteodes to reduce stress and to inhibit that Escherichia coli bacteria in the stable of these piglets. So this was quite interesting result. But after that, the guy was not very happy to. Well, we did it for 1 year with him, but the next year he was trying not to pay for that, and he had a problem with. Pedro had given to him a specific sequence to reduce flu because his piglets had a flu, and it worked well. And on the next series of piglets, he called Pedro and told, well, I have a problem. We have piglets ill and dying, and we broadcast the melody. What's wrong? And Pedro went to check and just realized that they had left a specific sequence for flu, which was not operating on Escherichia coli. So, he changed the sequence and the problem was resolved. But there is a high specificity of these proteodes. If you don't use the right one, you won't have the right effect, obviously.
Yeah.
On oysters, I told you this is not translated. Okay, but we had 3 aquariums. We compared aquariums. These were the results. We comparing aquariums with young oysters with those without proteodes. This is the blue curve. With one sequence of proteodes, you have the violin curve, and the yellow one is with the best sequence we had, and these are the level of the percentage rate of development of that virus, and the oysters without stimulation or inhibition, actually, of the virus were reaching the death level, you know, when the others were still completely alive. You had a small growth of the virus, even with proteodes, but the level remained well below the mortality level. And so you had at the end, after 10 days, 100% death with those having no proteodes and 0% with those having proteodes. It was very significant, but as I said before, we were not able to try to develop that on a wider basis. Okay, let's now have a look on market gardening, which may interest more of you. What we do is regulating seedlings growth and development improving the taste of fruits, increasing fruits conservation, preventing and fighting against virus and bacterial attacks, savings in inputs, water, energy, manure, and resistance to drought and freezing. That's quite a lot. Just a very old experiment on tomato culture in greenhouses in Switzerland. You see the difference with the greenhouse, the two above pictures. So the difference between the two greenhouses with proteodes and without proteodes, with the impact on growth and those without. I mean, they had the same quantity of water, but the only difference between the two batches were the proteodes. You can see that they are much better looking with the proteodes. And in the Senegal, you have also the tomato plants. You can see on the right that they are much higher with an anti-dryness music, and so they also had, which was measured, much more tomatoes. Well, they counted 10 times more, but well, it was not highly scientific, but there were really much more tomatoes produced with the same with less water, half quantity of water on the side with proteodes compared to the side without proteodes.
Wow.
How rapidly this slide should be elsewhere, but because I have it here, the company Genodics was created in May 2008. We have now nowadays a capital stock of 2,085 100 euro, but we also got additional financing from our shareholders, altogether about 680,000 euro. We are 4 full-time people and one external guy developing applications in vine in the eastern part of France. We have 62 shareholders that we grouped year after year. We were 5 when we began with Pedro and Joel and 2 friends, and after that we invited others. And the range of the investments of these partners are from 300,000 euro for the smaller, up to the smallest, to 50,000 euros for the 2 or 3 biggest. And we have this year 160 customers, about 300 installations. Our budget for this year is 350,000 euro, which is quite small after 15 years. I consider that we created a startup and we became a start slow up. But we continue and we hope that we will be able to find new investors this year. It would be the right time now after the COVID crisis to go up.
Yes.
Thanks. One very good point is that our customers are good customers. I mean, they trust us, and when they begin with us, we continue. And so this helped us during the crisis. We were not able to get many new clients. We were not able to visit new clients, but we continued working with our clients, which continue to pay us, and we survived to that crisis. So the fidelity of our customers is a very important point for us.
Yes.
Okay, let's come back to the market gardening applications. So, well, I already told that overlaps the previous slide. Just an example on an old application on tomatoes, where we measured the sugar contents, and we had the real differences between tomatoes with proteolysis and without proteolysis. You can see the rate of glucose and fructose and saccharose. The effect was mainly on fructose, which was doubled with music compared to those without the proteolysis.
So.
About prevention of diseases, we have experience on various plants, on salads with Bremia, Sclerotinia, and Rhizoctomia, courgette viruses, two viruses actually, and powdery mildew, tomatoes with botrytis and Rastonia, endives on Erwinia, cucumber viruses with CMV, CABYV, CYSDV, and 4-dinimellar form of cis, and strawberries with powdery mildew. We develop new applications when requested. Wow. An interesting work done when we began to work on endives during the phase of forcing. That producer had problems with Erwinia, this bacteria. Is it bacteria or mushroom? Well, Pedro should know that. But anyway, that pathogen, Erwinia, and in their forcing room, when they brought lots of new, the basis for chickens, and each day, and these were given to grow in obscurity without light during 21 days, and after that they took them away to take the mature chickens, and they measured so day after day they were taking off the production, having been 21 days in that room, and they measured the rate of plants, these endives, having problems second choice, they said. They had to remove leaves marked by that Erwinia. And due to the process of 21 days, we broadcasted the melodies during 21 days, and after that, 21 days without, and after, again 21 days with the melodies, and 21 days about. And so the sequential batches were getting from 1 to 21 days of music, according to the different batches. And you can see here in yellow the number of days of broadcasting, the specific sequence, and in red the rate of second choice of endives. And it's interesting to see the difference. I mean, it's clear when you see that on 3 months that you have a real effect of according to the number of days of broadcasting these melodies against that specific disease. So having seen that, that producer decided to continue, and she worked with us for 10 years, but she sold, she's a lady, she sold her farm last year, and to retreat, I mean, the guy who bought that didn't want to continue. That's one of the main causes of losing clients for us. When the client stops or sells his, or sometimes dies, or so, the followers don't believe in that and don't want to continue. But this was a very good client for 10 years. Other agroalimentary applications are growing fruits ripening. accelerating, improving, or regulating fermentation processes for better quality productions for bread, cheese, soya paste, or sake. In wine growing, we have good results in preventing Esca fungus. We reduce the mortality due to that fungus by 50 to 80%, an average of 2/3. On downy or powdery mildew, the effects are lower, but our clients learn to combine the traditional treatments and proteodes to reduce the quantities of treatments. And the advantage of proteodes being that they can broadcast all time when they see that the production is at risk, even when it rains, but they can't do with the products. And combining both, on an average, reduce by half the quantity of products they use on these diseases. We also have a reduction of 50% on short bud virus. Also on the phytoplasm named the golden flavescens, we have half less symptoms on the vines with broadcasting the corresponding proteodes. We have interesting results on root rot, on flowering, and frost, on against frost and drought. Against frost, we can lower by 1.5 and 2 degrees the level of resistance of the vine. It's not that much, but sometimes it makes a difference for low frost. You know, if you have what they say the dark frost with -7, 9, or 10, the protease won't be enough. But that's for the frost in spring. You know, when the flowers are beginning to appear.
Yeah.
On winemaking, we stimulate fermentation, alcoholic and malolactic, with success. For tree farming, we have experience on apples with cap fungi, frost, drought, and flowering. On peaches with plum pox virus. On vanilla with this began last September in Tahiti. Pedro was happy enough to spend 1 week in Tahiti to install these applications on vanilla, stimulating flowering and reducing the impact on Fusarium fungi. We also work in Corsica and with a big clementine producer, stimulating flowering, and the guy is very happy with the quantity of clementine that he gets, which improved. On flowers, we work on roses, high quality roses. We stimulate flowering and began last year to also reduce the rose dust. We are happy to work with a very high-level producer of roses used for the famous Channel 5 perfume near Grasse in southern France. We also had good experiments on gardenia and camellia, stimulating flowering and growth, and getting twice bigger flowers of these two plants with the same quality, the same contents level of interesting molecules. So twice the production of molecules used for perfumes with these two flowers was doubled.
Wow.
On the Charka virus on peaches, we worked on 120 hectares. In that case, with the broadcast equipment had to be over the peach tree, so the mast is 3 meters high. Pedro has to go with an elevator to install it and or repair in case of problem. This was one of the reasons, not the only one, to develop a broadcast model. An internet model that we can monitor through the internet. And now about 2/3 of our clients are equipped with that broadcast version, which allows to, the first interest for us was, in addition to the specific case, but was also to be informed quickly in the case of trouble. The box, all these connected boxes will connect to a server each evening, and if they don't connect, this means that there is a problem. So we are able to call the producer and ask him to have a look, and if necessary, we go to repair. But this also allows us to change the sequences according to the evolution of the needs of the plants, adding, for example, sequences against drought or heat when necessary, and stopping when not necessary. I mean, this evolution began 6 years ago, and was a very good thing. This is the beginning of an example. It was years ago, on apples against scab, and the guy is still continuing. This helped him to reduce by half the quantity of products used on that disease, and that's enough to be continued. So he has been working for 10 years and still working with us on that problematics with apples.
So dat is schurft, hè? Scabious. Okay.
Yeah. Yeah, that's the problematics needing the higher level of products to eliminate this makes, a gray gray marks on the apples, it's not toxic, but the customers don't like apples with gray marks on over this.
Michel, why is this not put on a high pillar like the former picture?
No, this was the beginning. Now it's higher. Yes.
Okay.
This is an orthograph. Yes, right.
Yeah.
No, this was the first test. I should change that. We have the photographs. It's now higher, obviously, like on the peaches. Yes, it is indeed.
Yeah. Okay.
Our project in environment would be to stimulate CO2 absorption, also stimulate the production of oxygen and biofuels by algae. We have an experiment showing that we can stimulate specific algae and have a much higher production of oxygen, for example, at the bench. We would like to regulate waste processing and also the production of methane by these big units processing the waste and helping to stimulate plants to depollute water. In pharmacy, we we know that we can predict specific functionalities of molecules, and we would like to stimulate the production of useful molecules for these companies. And about health, the potential is obvious, but request so-called clinical tests that we are not able to do. The costs are too high, and we cannot cannot afford them. When we saw that the COVID crisis in 2019, I was at the beginning very happy. I thought, well, that's a problematic for us. The viruses we can monitor quite well, and they said that there were no solutions, so we thought that we can help, and we contacted over 20 research teams in university hospitals, and got only 3 responses of people telling we don't have time to test that now. We can do it. And later on, when they had time, when the crisis resumed, we contacted again, and they said, no, we're not interested in that technique. It's not serious.
With all the restrictions, it must have been very hard.
But
Michel, I read somewhere that in the metros of London, these frequencies have been used to reduce the infection of people with COVID virus, and it seems to reduce the infection rate by 50%.
What did reduce by 50%?
Yeah, because of in the metros in London, they used some frequencies to fight against the virus of COVID, and it's really infection rate in
which frequency did they use?
I have to check. I don't know.
Okay.
Okay.
Well, I can tell you that we prepared. Pedro prepared a series of 8 proteodes to help with that disease, and we were very happy with that. We gave it to friends, but we are not able to implement widely. And I personally avoided the vaccine.
Yeah. Many in this group.
and I am happy with that.
Me too.
Anyway, so just to summarize, the Genodics services are problem analysis, looking for the key proteins involved, and selecting these proteins, and defining the modes of diffusion, how to set up and which broadcast protocol, installing the diffusion system, accompany the users, and transferring competences, and after-sales services. We always provide the maintenance service. Actually, we rent our broadcast boxes, and that includes the maintenance service. Our business model is to rent the broadcast boxes and to charge for a license for each application that we install with the producer. Well, the budgets are not very high. This is why we are not very big, but we should have more customers. The problem we face is, unfortunately, that chemicals products are cheap, and the producers know how much they pay to buy these products, and they are not industrial. They don't take into account the time they spend, the amortization of their machines, and all that additional factors. If they would do that, they would see that it's not that cheap to use these products. But they compare the direct cost of the products with what we charge for our box and license, and they say, well, we are quite expensive.
Then compared to all the costs for nature,
and obviously they don't have to pay. You're right. You're right. They should be charged for the cost for depolluting.
Yes, exactly.
The detrimental effects of their products. I fully agree. Okay. Just a last word on intellectual property. Steinheimer registered a patent in France in 1992, a very good patent, and then he tried to enlarge it at European level, and it took 40 years of discussion and additional experiments to validate it at European level, and it was finally published by the European Patent Office in 2007. And this is what which helped us to create the company in 2008. After having that European patent, we had that basic intellectual protection. But there is also an interesting thing, which is that we use musics, very specific musics, but they are musics. And they are also protected by the author's right, which is much cheaper than patent. It costs nothing. You just have to prove that you created that music and when you did. And you are protected in all 179 countries, which nowadays have signed the Berne Convention. And this covers the code, not only the music, but also the code and the methodology that Sternheimer developed to transpose the genome in sequences of musical notes, and even also of colors that we don't use. But there is also a set of proteochromies which may have also interesting effects. And that's for our protection. And the interesting point is that that protection is valid for 70 years for all the life of the inventor or creator, I would say in that case, and 70 years after. And we are happy that Sternheimer is still alive. So this gives us some time before us.
So you can
okay, that's it.
continue for 70 more years.
Yes.
Well, thank you very much, Michel. Wow.
So if you have
interesting.
other questions, we still have some time.
Are there any more questions from the others? I mean, there was also yeah, okay.
I have a question.
Okay, who is this?
Annette.
Annette de Jong? Nee,
Ja.
Annette van de Knaap.
Nee, Annette de Jong.
Annette de Jong, oh yeah.
Hey, Annette, hoi.
Hello.
So she's the one who has the...
yeah, Pedro installed this week the Genodics.
Yes, on last Monday, yes.
Yes. So our potatoes are happy now. My question is, we installed this for to make the potato plant stronger.
Yes.
And my aim is to use less chemicals and less fertilizer. So now we have a program for Phytophthora. And my question is, how can I immediately stop with the chemicals, or do I have to reduce it, or how? How can I measure this?
Yes. We know that problematics. We have other producers. You are the first one we work for potato seedlings, but we work on other vegetables with producers having the same problematics of reducing their products. I mean, it's just a question of prudence and being pragmatic. If you see that all conditions are in place for the development of a disease, we recommend to use some products. Because we long experimented on the mildew on various plants. First with vine and other plants after. And we saw that the years with a low pressure of mildew, which means that the weather was not too humid or too hot, the mildew developed little, and proteolysis was enough. But other years with better conditions for the mildew, you couldn't. Proteolysis were not enough. We had a test on a 7-hectare parcel of vine, and simulating against mildew. The producer kept 100 vines without treatment, and he got 10% of grapes only, you know. And the others he had treated, reduced, but enough treatment, and got a good production. So this convinced us to be prudent and careful, and not to recommend to stop the treatment. Okay. It's a question of observation, but we have a good client. We have been working with him for 8 years now, near Rennes in Brittany, and he tells us that I have a good interview of him made by a television company when he tells that he uses only 10% of the product he used before, and he's happy and will not stop the broadcast. But he's used to look at his plans day after day and see when there is a risk, and using the minimum quantity of products, and when music will be enough. So I don't have absolute recommendation. I just tell you to be careful.
Yeah. Okay. And then another question, because we grow seed potatoes, and seed potatoes must be free of Y virus, and they control in the field on the potatoes, and after harvest they do research in the laboratory if there's no tuber with the Y virus. Can you develop a program on that, on the Y virus?
I don't know that virus, but then you should tell it to Pedro. I'm afraid Pedro left. But what I can tell about viruses is that they are simple biological structures. As you probably know, a virus is a short sequence of a short genome having, according to the virus, from let's say 10 to 100 proteins only. So if you that compares with the human body having 23,000 genes able to produce proteins. The vine has even more, and you have other plants like rice, which can have 3 times more even. But so a virus having a limited number of proteins, if you select key proteins to limit his development and inhibit the production of these proteins, you will have a much stronger effect than with mushrooms or bacteria, which have thousands of proteins. And so, and that's interesting because plants are not able to produce molecules to resist viruses, antigens, as animals do, you know. So they are more at risk with viruses, and there is a very specific field of action for proteolysis against viruses just because of that. So, but we have first to know the virus which is at risk of developing, and then we will prepare. Pedro will look at its structure, choose the best, the key proteins to limit his development, and build the sequence and transfer it to your box if you ask him.
Yeah. Okay.
Okay. Any more questions, Annette, or someone else?
Yeah, I have a question.
Yes,
about the waste processing. You said that you
about
have
what?
Can you speak in the mic?
The waste processing. What?
Yeah.
You don't hear me?
You not.
That's okay.
Yeah.
Yeah. Okay. Okay. I have to look at the camera to be heard. You had one of the last slides. You talked about waste processing, and you said you reduced the methane production.
No. Just the contrary. We don't act on methane production. The problematics with these installations is to avoid H2S, you know, to have methane only, and so, but it's a soup of thousands of microorganisms, you know, and you have to be able to master to balance them in order to have a good decay producing naturally methane, and reducing the bacteria producing the other stinky gases. And so this is why what we would like to do, but we were not able to up to now to find a client to do that. We are sure we could help, but we didn't experiment yet.
Because in Dutch agriculture, this methane emissions from dairy farming and pig farming and chickens is quite important. So having a process of reducing that production or emission at least would be very helpful for more for future development.
Yes, but these needs requires test and adaptation. I mean, when we begin to work on the problematics, we rarely succeed fully at the first trial. It needs several experiments. For example, I spoke about oysters. This was only the 7th sequence which made the total effect. We had to test. We cooperated with researchers at the University of Caen and with that laboratory. They gave us useful information because we knew nothing about oysters and that virus. And so when they had understood what we proposed to do, they proposed us potential ways of actions and key proteins to be tested. And this is how we assembled the successful sequence. And that's the same for all problematics. For SK disease, we were very happy when we were informed of the problem in 2002. We asked a specialist in Switzerland who told us, if you can stimulate proteins, there is a key protein producing polyphenols and used by the vine to resist to that mushroom. And he gave it the name. The name is Caféoil coenzymase. Okay, he told us if you can stimulate that berry protein, you will have good effects. And we did it, and we had good effects. But we would not have guessed that alone. I mean.
Could you give a rough estimation of what would it cost if you want to develop this methane reduction thing? Would it be 50,000, 100,000 euros or more?
First it depends on the installation in place. And you have two aspects. You have the preparation work to analyze and understand and find the key proteins, and then practical experiments with measurement. The quality of the measurement of the analysis of the effect is important, obviously, to progress to the best possible application. I would say that the cost would be between, without to count for the analysis, which are usually done by the producer, between 20 and 50,000 euro to set up an operational application.
Okay. And there's also a question about what the cost would be for doing this in a dairy stable of 130 cows with focus on the production of fat and protein.
Production of what?
Fat.
Fat, fat
and protein.
Proteins, yes, fat and protein. Yeah, okay. Well, then the cost is not very high. The problematics we tried with cows, and the problematics was to analyze the part of the effect due to proteodes and the part of the effect due to the feeding and the other environmental factors of the cows, you know. And it was not obvious enough. We measured that there was an effect, but the part of the effect being due to the proteodes in comparison with the other aspects was difficult to assess, because in these cases you are not in laboratory conditions, and parameters may change from one day to another with more or less food, or change in the quality of the food, or variations on the weather, and so on. So it's well, the best result we had with milk was 30% improve in the quantity of proteins, but this one was on a very short time, and it was noticed after that that the cows got mammites, inflammation of so stimulating too much is not a good thing in any case.
Okay.
So you have to be careful. You can improve, I would
Yes.
say 10 to 20%, and it would you don't have to do it too much. So
Maybe I will bring Mike into contact with Pedro so that they might
yes,
see what's possible. Yeah.
Pedro has the experience. We did it with a farm in Normandy with cows, but they stopped because they decided to change the quality of the aliment, and they said it's too complicated to control for us, and so they didn't want to continue.
Okay.
Can you say already what it might cost, because you have the Proteode developed? Would it be
well, it's the question is only to stimulate the prolactin, which is the key protein for lactation. We have it, so it's not complicated to install it. Well, you will need to install broadcast equipment for automatic diffusion at specific times during the day and so on. But the annual cost will be the same as for potatoes. I mean, different Proteodes, obviously, but something about 3,000 euro per year.
Yeah, okay. Well, Mike, I'll give you the address of Pedro. And then there is one question. I think that would be the last because we have to stop. How do you relate genetics to homeopathy? Do you do that also or not?
No. I know homeopathy personally, and we also. I was happy to meet a long time ago the team working with Benveniste on what was called the Memory of Water, and so we became friends, and I helped them at that time to prepare their business plan to develop their activities, but unfortunately, short after that, Benveniste died of a heart attack, and the project has had to change. That work was taken over by Montagnier, as you know, Luc Montagnier, who continued. He recruited the key researcher from that team and worked for several years. We are investing a lot. They succeeded in having good publications, but not in getting clients. And so they had to stop, having spent too much money. And that key researcher created a new startup 3 years ago with a Swiss engineer. The name of the company is Life Signature, the last one. And they are still working, and we have plans to cooperate with them to see if water might have a role in the that memory of water. Is the water can memorize something about the effect of proteodes. We have science showing that it might be that there might be also a memory of water. Is everywhere, you know, in all organisms, plants, humans, or whatever, and it would not be a surprise for us that the water would contribute to the memory of the broadcast of this
topic.
So we have plans to experiment together.
Okay.
Good.
But that's different. That's different of homeopathy. I know the principles of homeopathy, but that's a different approach.
Yes. Yes. Well, we have some experts on water also in our network.
Oh yes, yeah. We also have contact with experts of information of water working in agriculture in France.
No, but it's not on homeopathy, which is of course always in water or in alcohol. But yeah, it's a different subject. Well, but maybe we should leave it at that. Annette, is it okay for you, that question? Yeah, okay. She says yes. Okay. Well, I think this was a very, very interesting session. And I'm very happy we recorded it so that we can look it up again. I will send it to everybody who has been here. And well, thank you very much.
Okay, thank you for listening. I noticed a few mistakes in my slides, so that I will correct.
We are used to the French English.
But I did that yesterday. I mean, a bit too fast, and some mistakes happened. But anyway.
But it's very nice that you did it in English and we could follow it.
Yeah. Michel,
thank you for listening, and I'm sorry not to speak Dutch, but
yes,
I'm happy that you can understand.
Yes. Okay.
Okay.
Thank you very much.
Merci beaucoup.
Okay. Merci beaucoup. And bye bye to everybody.
Cheers.
Okay. Bye bye.
Bye.